Hello friends, welcome to entranceadvisor.com. Hope you are doing well. So today I'll be discussing about the lecture 2, that is the chapter Biotechnology, the Principles and Processes. So let's start. Before starting, if you are new to this channel, please hit the bell icon and do subscribe our channel. So in the last lecture, I discussed about the genetic engineering. So what was that? The genetic engineering is the change in the phenotype or the expression of host organism due to interaction of an altered sequence of genetic material, that is DNA, into it. So that is genetic engineering is the biotechnology involved in the synthesis of artificial genes, repair of genes through fusion, deletion, inversion, shifting of genes, and the production of the recombinant DNA, that is the combination of DNA from two different sources and manipulating them for improvement in human beings, plants, animals, as well as in microbes. An important aspect of genetic engineering is recombinant DNA technology or the recombinant DNA combination to give a desired product or a desired trait. It is also called gene splicing as it involves cutting and pasting of the desired DNA fragment. It is based on two important discoveries in bacteria, that is the presence of plasmids and the presence of restriction endonucleases. I'll be discussing about that shortly. So coming to the recombinant DNA technology, which is also called genetic engineering, deals about the production of the new combinations of genetic material artificially in the laboratory. This recombinant DNA or DNA molecules are then introduced into the host cells, okay, where they can be propagated and then multiplied. Now there are some steps of the recombinant DNA technology. First one is identification of DNA with desirable genes. Next one is introduction of the identified DNA into the host. Third one is maintenance of the introduced DNA in the host and transfer of DNA to its progeny. There are several tools of recombinant DNA technology that includes restriction enzymes, polymerase enzymes, ligases, vectors, and host organisms. So recombinant DNA technology is the field of biotechnology that specializes in developing new combination of the genetic material called the recombinant or the rDNAs to be artificially constructed in the laboratory for the introduction in the host cells for the propagation and multiplication. The various tools required for the DNA, recombinant DNA or rDNA technology are the enzymes, that is the source of donor DNA, polymerases enzymes, ligases, vectors, and the host organisms. So what are the restriction enzymes? Coming to the Albert discovered that Escherichia coli or E. coli in 1963 is able to protect itself from the attack of the bacteriophages with the help of a defense mechanism called the restriction modification system, okay, which has two components. First one is the restriction enzyme. The enzyme is a nuclease which recognizes a particular palindromic DNA sequence that and degrades for the same. It was called a restriction enzyme because it restricted the propagation of the bacteriophage, that is the foreign DNA in the bacterium. Second one is methylase. The enzyme adds methyl groups to particular palindromic region of the bacterial DNA so that the restriction enzyme is unable to harm the bacterial genome. The tools, as I said, it's written here again, and hence I was saying that restriction enzymes are also known as molecular scissors. The restriction enzymes are the DNA cutting enzymes. Each enzyme recognizes one or a few target sequences and cuts DNA at or near those sequences. Many restriction enzymes make staggered cuts, producing ends with single-stranded DNA overhangs. However, some produce plant ends. DNA ligase is the DNA joining enzyme. If two species or pieces of the DNA have matching ends, ligase can link them to form a single unbroken molecule of DNA. In cloning or the DNA cloning, the restriction enzymes and the DNA ligase are used to insert genes and other pieces of DNA into plasmid. As you can see, the recombinant DNA molecule is there, and here is a vector, and there is a fragment of DNA. With the help of the ligase or DNA joining enzyme, it is ligated, and this cut was made by recombinant DNA molecule was these cuts were made by the molecular Caesar or the restriction endonucleases. And then the whole process of cloning continued. That is, the bacterium was there, then it is transferred to a host cell, then the bacterium carrying this recombinant plasmid or DNM, and multiplication of the recombinant molecule takes place within the host. Here it is bacteria or E. coli. The division of the host cell takes place, as you can see, the numerous divisions resulting in a clone. Clone means similar type of colonies, bacterial colonies were growing on a solid medium or agar. Okay. Then you can see this restriction enzymes are the DNA cutting enzymes. Each enzyme recognizes one or few cutted and DNA and cuts the DNA at or near those sequences. So many restriction enzymes make staggered cuts producing ends with the single-stranded DNA overhangs. However, some produces plant ends. The DNA ligase, as I said, is the joining enzyme. If the two bases matching ends, then it can link. Here it's written multiple times just to demonstrate you a bit more. And restriction enzyme is also called the restriction endonucleases or REN. The first restriction endonuclease discovered was Hindi2 from the Haemophilus influenzae by Smith. Up to now, some 900 restriction enzymes have been isolated from over 230 strands of bacteria, and the enzyme name has the first letter of the bacterial genus, next two letters from the species, and the fourth letter from the strain, if any. The example, the E. core R1 or the Escherichia coli RY13 have the last Roman numerical signifies the first endonucleases from the bacterium. However, the Arbor and Smith and Nathans were awarded Nobel Prize for that in 1978 for discovery of the restriction endonucleases. Cut the strand of DNA a little away from the center of the palindromic site. What is palindromic site? You will get to know a bit later. And between the same two species of the opposite strands having sticky strands. The stickiness of the strand facilitates the action of the enzyme DNA ligase. The restriction endonucleases are used in the genetic engineering to form the recombinant molecules of the DNA, which are composed of DNA from different sources of genome. Okay, when cut the restriction enzyme, the resulted DNA fragments have the same kind of sticky ends and can be joined together by using DNA ligases. As I've told you earlier, that they are nuclease enzymes. These there are two types of nucleases. Okay, one is exonucleases and one is endonucleases. Exonucleases cleave the DNA or RNA molecule from the ends only. Okay. Suppose this is the DNA molecule. You can see. Sorry. This is the DNA molecule. You can see. These are the ends. If the restriction enzyme cut here, then it is obviously it's our exonucleases. And you can see the E. coli cuts in the middle of the DNA strand, making a nick. So it is an endonuclease. Okay, the endonucleases cut the DNA molecule from inside. The restriction endonucleases cut the DNA at the specific sites called the recognition sites. Okay, this site is called as restriction or recognition sequences. Each restriction endonuclease scans the DNA sequence. When it finds its recognition sequence, it sticks to that and cuts each of the two strands of the double helix at specific points through their sugar phosphate backbone. So you can see that these are the steps in the formation of the recombinant DNA or rDNA in the restriction nucleases by EcoR1, and the steps you can also find it in your book itself and other books. So The sticky ends are joined by the DNA ligase, forming a recombinant DNA. Here, the recombinant sequence of each restriction endonucleases is a palindromic sequence. Same. In the palindromic region, the two strands of the DNA possess the same base sequence but in opposite fashion, that is 5' to 3' and 3' to 5' directions. It is just similar to the palindromic word which reads the same in both the direction. You know, the Malayalam or Ramar or etc. Okay. Uh, the restriction endonucleases attaches itself between the same two bases of the opposite strands and causes a break in the two. Because of it, the restriction endonucleases are also called molecular scissors or chemical scalpels. There are two groups of restriction endonucleases. One group of endonucleases produce short single-stranded ends which can join single-stranded ends of the other DNA molecules or fragments having the complementary sequences. Such ends are called staggered ends or sticky ends or cohesive ends. E. coli from Escherichia coli or other bacillus belong to this category. Now, so in this you can see it is a separation and isolation of the DNA fragments taking place, and the fragment of the DNA obtained by cutting DNA using the restriction enzyme or R enzyme is separated by a technique called the gel electrophoresis. Negatively charged, I will show you this process how it looks like. The negatively charged DNA fragments can be separated by forcing them to move towards the anode under the electric field through a medium. Under the DNA fragments separate according to their size through the sieving effect provided by the cross gel. And the separated DNA fragment can be visualized after staining the DNA with a ETBr or Ethereum bromide followed by exposure to the UV light. Separated bands of DNA are separated from the agarose gel and extracted from the gel is called dilution. The DNA fragment purified this way is used for recombination. So the foreign DNA, the same restriction is then cut for foreign and the DNA at specific point. This is their R side, and the ligase is joined to form the foreign plasmid in the transformation and the cells divide. Yes, here it is. The typical agarose gel electrophoresis showing the migration of the undigested len and digested set of DNA fragments. This is the actual electrophoresis of the genomic DNA. Okay, these are the bands of the DNA. You can see these are just the bands in different lanes. Undigested DNA is represented by sharp border near the walls of gel, while smearing indicates the digested DNA sample. You'll get to know in future a bit more. Now coming to the cloning vector, if the DNA forms, if the DNA from the various sources or different sources are cut by the same restriction endonucleases, they would produce the complementary of the sticky ends. With the help of the DNA ligase, the complementary of the sticky ends of the two DNA can be joined end to end to create the recombinant DNA. You got it? I think you got it, what I am saying. So, after the separation and isolation of the DNA fragments was done by the gel electrophoresis process, as I have shown you, then coming to the cloning vectors, as given to your in your book, and they are the carriers. What are the cloning vectors? Okay, they are the carriers or the vehicles of the desired DNA fragments of the passenger DNA which can undergo independent replication to increase the copies of the desired DNAs or genes, plasmids, that is plasmids, bacteriophages, etc. The number of plasmids in some bacterial cells can be 15 to 100, even higher, through the common number 1 to 2. Similarly, a number of bacteriophages mature in a bacterium. Therefore, if we are able to recombine the plasmid or piece of DNA with the bacteriophage genome or a plasmid and introduce the same in a plasmid-free bacterium, the vector would multiply and equal to the number of plasmids or bacteriophages that normally develop in it. So here you can see the plasmids and the bacteriophages commonly used for cloning. Okay. Colonies is the same kind of colonies of growing of colonies, bacteria. Here I'm talking about, and they have the ability to replicate within the bacterial cells. so independent of the control chromosomal DNA. The bacteriophages, because of their high number per cell, have a very high copy number of the genome within the bacterial cells. The following features are required to facilitate a cloning into a vector. That is the origin of replication. So a cloning vector or the passenger DNA can be of three types, that is complementary, synthetic and random. It must have its origin of replication, a selectable marker for identifying oocyte, selectable marker for identifying the recombinant form of non-recombinant and preferably a single recognition site for cloning. The cloning vectors are of four types: plasmid, viruses, cosmids, and the artificial chromosome. The plasmid vector for the plants are different from the others. Here it is. Origin of replication, it is the sequence from where the replication starts, and any piece of DNA when linked to this sequence can be made to replicate within the host cells. This sequence is responsible for controlling the copy number of the linked DNA. Now coming to the second number, of the selectable marker, helped in the identifying and eliminating non-transformants and selectively permitting the growth of the transformants. And the transformation is a procedure through which a piece of DNA is introduced into the host bacterium. Generally, the gene code encoding the resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, or kanamycin, etc., are considered useful selectable markers for the E. coli. Now, coming to the cloning sites, to link the foreign DNA, the vector need to have a single recognition sites for the commonly used restriction enzymes, as presence of more than one recognition site within the vector will generate several fragments, which will complicate the second cloning or the gene cloning. The ligation of the foreign DNA is carried out at a restriction site present in one of the two antibiotic resistance genes. And at last, the vectors for cloning genes in the plants or animals, the Agrobacterium tumefaciens. The pathogen of the dicot plant is able to deliver a piece of DNA known as tDNA to transform the normal plant cells into a tumor and direct these tumor cells to produce the chemicals required by the pathogen. Retroviruses in animals have the ability to transform the normal cells into cancerous cells. The tumor-inducing TI plasmid or the Agrobacterium tumefaciens have been modified into the cloning vector, having no more pathogenic to plants. Similarly, retrovirus have been modified into cloning vectors for animals. So, the plasmids are what are the plasmids? The plasmids are the small extracircular, you can see, extracircular DNAs which carries the extrachromosomal genes in the bacteria and some fungi. They replicate independently. The best known vectors which are also available commercially are PBR 322, as you can see, and PUC18, PUC. Here it's not drawn over here, but you can see the PBR, its most common example showing the restriction sites over here, that is Hindi3, E. coli, EcoR1, BAMH1, SALA1, PVU2, PST, and etc. over here. The best known vectors are those, and both of them have been modified from the natural plasmids of the E. coli. And PUC series of the plasmid vectors are named so because they were initially developed at the University of California. So let it be. They have derived from the PBR 322 itself, and the plasmids are about 2700 or 2.7 K base pairs long. They have the common or the following features, as I've told you, that is the origin of replication or ORI site. It is the DNA sequence which is specialized to initiate the replication. The series also possesses nearby replication control, which determines the number of the copies it would form. Therefore, the selected plasmid should have an origin of replication that supports a high copy number. Okay. Now coming to this selectable marker regions, that is the AMPR and the TET-R. Here the plasmid has a selectable marker. Okay. Normally the gene encoding for the antibiotic resistance, that is ampicillin and chloramphenicol, tetracycline, kanamycin, are considered useful as a selectable marker for E. coli. And PBR 322 has two resistance genes, or ampicillin resistance or the tetracycline resistance, while PUC18 has only ampicillin resistance gene only, let it be. And selectable marker is meant for the distinguishing recombinant from the non-recombinant DNA. It also helps in eliminating the non-transferred bacteria non-transformed bacterial cells from the transformed bacterial cells. Okay, what is the transformation? It is a passage of the foreign DNA or the plasmid containing a passenger DNA into a host cell. I'm showing you what it is. That is insertional activity. So by this process, I'll show you. The cloning site or the restriction site or the site where alien DNA can be inserted should preferably be single and responsive to commonly used restriction enzymes. Okay, the occurrence of more than one recognition site will cause DNA fragmentation and disrupt the gene cloning. Okay, in PBR 322, the alien gene DNA is ligated generally in the area of BAMH1 site and tetracycline resistance gene site. The recombinant plasmid does not possess the tetracycline resistance but continues to have the ampicillin resistance. The plasmids are therefore passed into the bacteria during the transformation. The non-recombinants can flourish in the medium having both ampicillin and tetracycline. Recombinants can grow only in the ampicillin containing medium but will die out in tetracycline containing medium. And this activation or the insertional inactivation of the gene due to the insertion of the alien DNA is called the insertional inactivation. Okay. The plasmids with antibiotic resistant genes are as selectable markers required to follow a cumbersome procedure requiring antibiotic containing culture plates. Therefore, the alternate selectable markers have been produced and they use the chlorogenic substance, that is the XGAL, that is the coloring thing, and the common recognition for or the cloning site is LAG-C or LAG-Z that codes for the enzyme beta-galactosidase. Insertion of the alien DNA into it inactivates the gene and prevents the synthesis of the enzyme beta-galactosidase. The chromogenic substrate produces blue-colored colonies. If the plasmid do not have inserts, the white-colored colonies in the recombinant plasmids are present. You can see clearly that here are various blue and white colonies, how it looks like, and it's the XGAL which is making the color, and which is get inserted into their plasmid, their DNAs, and forming the beta-galactosidase to express themselves. So the most efficient method of the screening for the presence of recombinant plasmid is based on the principle that the cloned DNA disrupts the coding sequence of the gene. This is termed as insertional inactivation. For example, a powerful method of the screening for the presence of recombinant plasmid is referred to as the blue-white selection. This method is based upon the insertional inactivation of the LAG-Z present on the vector. The LAG-Z encodes for the enzyme beta-galactosidase, as usual in general, okay, which can cleave the chromogenic substrate into a blue-colored product. If the LAG-Z is inactivated by the insertion of a target DNA fragment into it, the development of the blue color will be prevented and it gives a white-colored colonies. By this way, we can differentiate the recombinant white color and the non-recombinant blue colored colonies. I think you got this idea. Now coming to the competent host. What is it? For the transformation with the recombinant DNA, the simple chemical treatment with divalent calcium ions increases the efficiency of the host cells through the cell wall pores to take up the rDNA plasmid. rDNA can also be transformed into the host cell by incubating both and ice, followed by placing them briefly 42-degree heat shock and then putting them into back ice. Then it enables the bacteria to take up the recombinant DNA. As I've said, in microinjection method, the rDNA is directly injected into the nucleus by using a glass micropipette. And ballistic or the gene gun method, it is also known as has been developed, or it is also known as particle gun method, or it has been developed into and introduced rDNA into the mainly plant cells, causing a gene or particle gun. In this method, the microscopic particles of gold or tungsten that are used are coated with the DNA of interest and bombarded onto the cells. And these are the only, and there is another process known as electrophoresis, which is not mentioned here, but you know that. And the last method uses the disarmed pathogen of the vectors or agrobacteria mediated, that is a natural way of vector transformation, where and when allowed to infect the cell, the transfer of the recombinant DNA into the host takes place. Okay, I'll show you in the next lecture, if possible, what is this microinjection means. That is this telling you about the electroporation method. That is making the cell membranes porous with the electric current for the entry of the desired genes or the other genes or the other molecules. Okay. And competence, what is competence? Competence is the vector transfer method. It is the ability to pick up the genes and the plasmid from the outside. The DNA is hydrophilic molecule. It cannot pass through the cell membranes. Okay. And however, the competence can develop for a short duration towards the um, end of the growing period. The competence can be enhanced by treating the host cells with a specific concentration of the divalent ion, like the calcium and electrophoration, that is the electric current. It stimulates the attachment of the DNA fragments or the vectors over the receptor sites of the cell wall. The cells are then incubated on ice by a brief heat shock at 42 degrees Celsius and again putting back on ice. As I've told you, that is heat shock method. The foreign DNA or the particles passes through the plasma membrane and enter the cell. It is known as the electroporation technique. And there are the other methods, that is the direct method without any vector or vectorless transfer. That is the desired genes as well as recombinant can be passed into a plant or animal or human cells through a microinjection technique. That the thing looks like this. This is the holding pipette and this is the cell. Here the glass or microinjection is injected with a needle with the DNA. The DNA is inserted with this microinjection, and this one is nucleus. Inside the nucleus it is done. Next, the particle gun or particle or gene gun or ballistic gun method, where the tungsten or gold particles are coated with the desired genes are bombarded into the cells with a great force. Instead of special sprays are also used for this process. Here are some differences. You can look over this. You can pause this video and write in your copy for extra information. I have just put that in a way. What are the differences between you, and it is important for your exam too. Most probably this and this. What are the differences? And I hope you enjoyed this video. So thanks for watching. Take care.